Hi everyone, now in this video, we are going to talk about a common tested exam question about rate of photosynthesis. So let's dive into it. So whenever you see that there is a lamp as well as a mixture that contains chloroplast, now the understanding is that anything related to plants and a light will always boil down to the concept of photosynthesis. Okay, so in this case, we are looking at the rate of photosynthesis. Now, definitely, can you see there is a ruler over here? Now, what is the idea behind putting a ruler over here? It is to measure what? The distance, right? The distance between the lamp and the mixture. And you see how they will actually result in the time taken for the mixture to turn green, or how long it would take for photosynthesis to occur. Now, can we look over here? They say that they are going to record the time taken, and then the experiment was repeated with 3 different tubes. Okay, so now first off, we must understand who is the change variable. Whenever we conduct experiment, we will always have a change variable and the measure variable, right? So in this case, can you see definitely the change variable will be the distance from the lamp, okay, the light intensity, right? Can you see from here to here, what is the amount of light? Okay, from here to here, there's definitely going to be less light, right? So when you change the distance from the lamp, you are doing a changing of a variable, right? So that is the change variable. Now, what is the measure variable? Now, the measure variable is the time taken for the mixture to turn green. That also means that the chloroplast has already taken effect, right? Undergone photosynthesis. Okay. Now, so what you can see over here is that when the distance is the smallest, the time taken will also be the smallest. And when the distance is the largest, the time taken will also be the longest. So can you see over here, the idea is that when the light intensity is very high, when it's close to the lamp, what will happen? Photosynthesis will be fast. Okay, when this happens, distance is far, less light, photosynthesis will be slow. Okay, now, so let's look at this. They tell us the relationship, the time taken for the mixture to turn green increases as the distance from the lamp increases. So now just now we have to look at this question is always a command word, right? Command word is explain why. Now, the variable is the distance from the lamp increases. Now you must always talk about the amount of light, right? Or the light intensity. As the distance, now this is the change variable, I say again. Okay, so as the distance from the lamp increases, okay, now we need to talk about light intensity, right? So light intensity is definitely going to go down, right? Light intensity will decrease, all right? And you need to pull it back to the question. The question says the time taken for the mixture to turn green increases. Now, you have not answered why the mixture is going to turn green, because of the fact that photosynthesis occur, right? So when light intensity decreases, hence the rate of photosynthesis is also going to decrease. All right, so I've brought you through how to answer part A, which is a very typical question about relating the distance of a lamp to the sample and also talking about how that impacts on the light intensity. Okay, now next, in part B, now they say that the experiment was conducted in a dark environment. Okay, now give a reason why this made a fair test. Now, fair test must always be what? Let me revise together with you. Fair test, you must only remember that a fair test can only happen when there is one change variable, okay? And the rest will remain constant variable, okay? Or all of them will remain controlled, okay? There will be constant variable, okay? Now, so how do we answer this question? It is always CV affect the MV, right? This is what I have told you all this while. So that means to say, now, whenever we answer this, we need to write down, it is always to ensure that keyword only the change variable. Who is the change variable? The distance from the lamp affects the MV, okay? Affects the rate of photosynthesis and, okay, only the distance from the lamp and that the lamp is the only light source, right? Okay, Okay, so I need you to be able to see this for part B. And now let's look at part C. Now they say that John repeated the experiment with a fourth tube W. Now W had a different amount of chloroplasts. Okay, so when you have a different amount of chloroplasts, now definitely we are using a new change variable, right? Can you see now the amount of chloroplasts is going to be the new change variable? Okay, now he placed W at a distance of 10 cm. Okay, from the lamp, the time taken for the mixture to turn green was 5 seconds. So how can we answer this question? Which one had a larger amount of chloroplasts? Okay, now definitely W, right? Why? Because if you look at the data over here, now comparing with S have amount of chloroplasts that require 9 seconds to turn green. But if you have more chloroplasts, definitely it will turn green in a shorter time, right? So W had a larger amount of chloroplasts. Okay, since since okay, the time taken was shorter. Okay, now the larger amount of chloroplasts, what do? They allow more light to be trapped so that the rate of photosynthesis will increase, right? So allow more light to be trapped, okay, increase rate of photosynthesis. So I hope that you are able to see how this question ties in with the concept about photosynthesis, okay, and I hope that this question is you are able to do it independently.